This is episode 18 of the Immunology Podcast, Immune Microscopy with Dr. Phan. Hi everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today we have Dr. Phan from the Garvan Institute of Medical Research and the University of New South Wales on the podcast to talk about his research using intravital two-photon microscopy to track the origin and fate of cells' critical immune responses. Yes, we do deep dive into immune microscopy here, and as a fellow microscope nerd, we got very, very nerdy in all the good ways. We've also got our usual roundup of recent highlights immunology news coming up, but first...
STEMCELL is hiring. STEMCELL Technologies is a world leader in developing services and tools for scientists working in cell biology, regenerative medicine, immunology, cancer, and disease research. United by a foundation of scientific integrity and driven by a mission to advance science globally, STEMCELL is a team of scientists helping scientists. They are looking for creative, driven people to join their international team. Explore more than 60 current offerings in areas such as R&D, sales, business operations, quality, and science communication at jobs.stemcell.com.
Well, Brenda.
How are things over there? Is the turkey already purchased for Thanksgiving?
I don't know because it's at my in-laws and I don't have to worry about it. And it means I get a 4-day weekend next weekend, which will be most excellent.
Well, good for you. We do get some, Black Friday offers here, so I guess that's good. Yay, consuming.
Yeah. The fact that you guys have Black Friday as well is kind of weird. But
we do not go all crazy and just camp outside our version of Best Buy to get a new TV. We just order stuff online mostly, I'd say.
There's not an injury on Black Friday making the front page of the news. It's not Black Friday.
Indeed. We have European version, a little bit more civilized.
All right. I'll start with my COVID paper du jour, which is titled Identification of a Therapeutic Interfering Particle, a Single Administration SARS-CoV-2 Antiviral Intervention with a High Barrier to Resistance. It's in Cell, accepted the 2nd of November. It's still in the pre-proof mode online. First author is Sonali Chaturvedi, and the last author is Lior Weinberger. So this is an interesting paper because I learned a lot about virology to do this. So viral replication is pretty inaccurate. And sometimes when a virus replicates, it makes a form of itself that inhibits its own replication. So that's like a known natural phenomenon that happens, but there's been a question for a long time of whether this could be used as a therapeutic by creating a partial version of a virus without necessarily all the things in the middle that you dope in. The virus will naturally replicate it because it has the 5' up, the 5' UTR and the 3' cap on it. So it looks like it. You cut out a lot of the juicy middle, maybe throw a reporter in for good work. But it's replication incompetent. So it replicates, but it soaks up the replication cycles of the virus's proteins to make a bunch of it, not a bunch of the actual virus. So this is called a therapeutic interfering particle, a TIP. And long story short is they made a TIP for COVID, for SARS-CoV-2, and showed that it works, essentially. But then there's features of this which get really important, that if it's too interfering, it won't replicate itself and will kill itself out. So it can't be so interfering as to completely prevent viral replication processes, because then it won't make more of itself. But it has to be kind of this right sweet spot. And so they kind of do like a shotgun system where they have like 2 or 3 different ones that they go with and then look at how they work and kind of optimize based on known computer modeling to get the ones that they use in the study. And of course, then they pick the best one. So you kind of want this particle that will jump in line pretty well to get replicated, but not so well that the virus doesn't do any replication. And they show that in In a variety of systems, in vitro, in vivo with hamsters, it reduces the infection viral load too long. So 100-fold. That's pretty cool. I'm going to talk about delivery systems, which could be lipid nanoparticles. In fact, I mentioned intranasal lipid nanoparticles as their mechanism, which kind of goes back to our talk about IgA and intranasal administration of medication. And through a lot of work, they show that it indeed can work. And then they actually look and make sure that there's not a mutational resistance here. And so the virus can't just mutate out of this, which it doesn't. There's also some concern that these types of products could push the virus to evolutionarily adapt and become more dangerous, because it's creating a pressure on the, selective pressure on its replication processes and sensitivities. That's still an outstanding question because they just don't have enough data from this paper. But they do show that after many passages with this co-culture, it doesn't develop some like super virus or develop resistance to it quickly. So there you go. You can take like the ends of the viral RNA, stick them together with a little bit of stuff in between, and make a product that inhibits regular viral replication by being basically a the pseudo-ribosomal sink.
That is really cool. I never thought of. I was not aware of the existence of these viral particles that could interfere with the replication of the actual virus. I think it's so exciting now also that we have more and more different ways of therapeutic molecules or therapeutic products to help reduce the severity of COVID. My question is, like, do they discuss if this would be applied therapeutically, for example, through a nasal spray or something like that? What is the timeline that they should be administering?
So they look at even administering it a day or two after known infection, and it works because it drops the replication. So you want it to be like really bad. You want it so that the virus, the RNA polymerase will work decently well on this, but the human ribosome doesn't make anything useful out of it. And so it's been the human ribosome that's been hijacked will go and try to make a bunch of nothing.
Right.
But the RNA polymerase can't like get stuck on it. And so you're not replicating the virus anymore.
That's very cool.
Yeah. It's pretty neat.
And are they like planning on moving this forward? Do they?
I think they're unsure. It seems very proof of concept. I think there are some concerns about like the evolutionary biology here of shoving in something that is this unknown, but we'll see.
All right. That's very nice. But then it's a good kind of segue into my COVID paper, because why have one when you can have two?
Well, why have 1 year of pandemic when you can have 2 years of pandemic?
Well, there you go. The rules just keeps applying. And this paper has been quite popular in science Twitter. Because it identified very nicely kind of non-spike directed immunity that results in a kind of aborted SARS-CoV-2 infections in highly exposed individuals. So what do we have here? We have a study done from the lab of Mala Maini at University College London. A bunch of 6 first authors, so a big collaboration here. And they focused on a group of people, which are most of, in this cases, health workers that are known to have been exposed to SARS-CoV-2 and have many of their colleagues tested positive by PCR, have a positive serology against SARS-CoV-2, but they don't develop any kind of detectable infection by PCR or by serology. So the question is, what are these people? How are these people preventing infection? And why don't these people develop immunity, kind of humoral immunity like most people do? And so what is really cool, so they look at this, they have samples from healthcare workers, some blood samples from before, some blood samples throughout the beginning of the pandemic, and they show that these patients are in a cohort of people. They are, as I mentioned, healthcare workers, that are very much exposed, and most of these workers do exhibit infection, but this group doesn't. They show that it's very rare not to exhibit seroconversion after being PCR positive. So it really does seem that these people that have not really developed a detectable infection. And when they start looking into their blood, so they say, well, if we don't see any B cell responses, what about the T cells? And this is really, really cool. They show that these people actually do have a response, a T cell response against non-structural proteins, mostly around what they kind of call the replication transcription complex of SARS-CoV-2, which includes RNA polymerases and helicases that are related to viral replication. And these are not as highly expressed during infection as the structural proteins, which makes it very interesting to understand why are these proteins being targeted. So they show that these people do have a higher level of T cells that are responding by ELISpot to these antigens. And they also show by using a marker that they previously described, which is an interferon inducible gene, which is IF27. And they show that in their previous work, they show that IF27 is positively related to COVID infection, and they can actually, this marker comes up in blood earlier than actual PCR positivity. And they show that the seronegative patient or the seronegative healthcare workers actually have a higher IF127 baseline, which suggests that they actually have had an infection ongoing that was mediated by, which the response was mediated by T cells. So basically what they show is that these patients are actually aborting a full-blown infection by very early on picking up the virus through the expression of this other group of antigens. It is not clear to them whether the reason these healthcare workers have this immunity is because they are either have been exposed to other coronaviruses. There are some level of conservation of these proteins between coronaviruses more than for the structural proteins, or whether these people, by being frequently exposed to very low levels of SARS-CoV-2 because they are wearing all this protective gear, but they're kind of constantly exposed, maybe this allows this T cell response to generate without a full-blown B cell response accompanying it. So I thought it was really cool because there was also other papers a lot earlier in the pandemic that showed that there were some pre-existing immunity against COVID. And I think this is probably the similar idea. Probably are kind of both these studies are looking at the same thing. And it is interesting to understand whether it is due to other coronaviruses or it's because of this baseline exposure that allows the development of the T cell responses in this people. So it was very cool. And I think, it was a very popular topic lately. And it was a really nice paper to look at.
So did they find any genetic differences in those people who had this early response or not, or did they not look there?
No, they don't look into what do you mean, like SNPs or stuff like that?
Like host genetics of any form.
No, they don't go into that.
Okay.
They look into other things, just age and group, but they don't see any other specific markers.
And just to be clear, T cells get the job done.
T cells get the job done even before B cells have the chance to react to this antigen. So yeah, I'm not surprised, I have to say.
All right. Well, we'll have to move away from COVID now, unfortunately, but not unfortunately, and go to something completely different, molecular dead man switch for viral infection. So I guess still COVID. but not really. So taking a high-level view here, there's something called the guard hypothesis that this paper just spends some time describing. And I'm gonna explain this before I go into the paper. And this is the idea that a host cell that's infected by something has another mechanism that notifies itself that it's been infected, right? So it monitors or guards critical paths so that disruption of those paths by the virus or virulence factors provokes an immune response. Okay, so when a virus develops an evolutionary escape mechanism or evolutionary mechanism to, say, turn off your cytokine signaling, it senses that the fact that its cytokine signaling is turned off and then does something. So this is a paper about self-guarding. So now to dive in, it's called Self-Guarding of MORC3 Enables Virulence Factor Triggered Immunity. This is by Moritz M. Gates, first author, and last author is Russell E. Vance, and it came out in Nature on 10th of November. So in this case, they just diving right in, they show that MOCR, which I'm going to try to find it because they only say the name of it once. Let me get it in here. So it's a nuclear body protein, which they don't actually specify what it stands for. That's why I was having trouble finding it. So the way it works, though, pretty simply, interferon is a main mechanism by which viral mediated immunity is managed. And certain viruses, in this case herpes simplex 1, tries to modulate interferon production. So it has something called ICP0, which targets proteins for degradation through the SUMO pathway. And in doing so, it drops down interferon signaling generally. However, this paper shows that there's a counter effect that's a self-guard. So MOCR inhibits interferon production. And how it works is that this virulence factor in herpes, ICP0, causes MOCR to be degraded, thus leading to activation of interferon. So the virulence so it's a self-guard in that the virus goes to degrade this protein as part of its function, and in doing so auto-generates the interferon response against it. So it's like this double whammy negative regulator. I think this is one of the first examples of a self-guarding being discovered, which is why it's a big deal. And it's independent of the STING and IRF3 and IRF7 pathways. So double-stranded DNA sensing, it's not through that. This whole SUMO-mediated ubiquitination and degradation process. And MOCR happens to be a target of this ICP0 protein that does this, and that then leads to the activation of the very pathway the virus is trying to turn off and kills the virus. And so they do all the knockouts to show that it's important and it works this way. And if you get rid of MOCR, you don't have this response, and then the virus doesn't die. But other pathways, if you block like STING or what have you, you can see different effects because they show that they're independent. So they do all the knockout work, show that they are really independent pathways, and basically put it all together in a pretty succinct paper. It's not a large paper. It's only 5 pages. And they also show that this MOCR, there's a regulatory element, MRE, and it's location dependent because all the genes that it seems to activate are on chromosome 9. And that drives interferon signaling all in that loci. This is not true of other cells. This is not true in embryonic stem cells. It is only true in monocytes that they found. So there you go. Apparently we have a molecular dead man switch through a pathway that the viruses attempt to inhibit, downregulate proteins as a virulence factor actually causes its own death.
It's funny that it's monocyte specific. What I guess that doesn't make sense to have this in other cell types is because monocytes are particularly susceptible to infection with this virus, and thus they need some extra lines of protection.
So I don't think they went through T cells and other stuff. on here, but they went through mature immune cells that could be infected by HSV, such as monocytes, and I think it's a cell culture question as well.
Okay. Are they using monocyte cell lines?
They are.
Well, it's nice and ironic that the virus is doing all this work to stop the interferon response, and then it just doesn't get to do it.
Right, because
redundancy is very important, clearly.
Yep, and then the other targets of the SUMO pathway, because it degrades SUMO-dependent IF-1, 16, and ND10 nuclear bodies, so it blocks. So this ICP0 pathway that the virus has, this virulence factor, does block other downstream things from being affected. So it does block the other interferon, but it then causes this other thing to die to release interferon. So you're damned.
And that is the purpose of that is the purpose of ICP0, right? To block these interferon pathways.
Yes. It just then happens to block a protein that blocks the pathways, or degrade a protein that blocks the pathways.
Okay.
Oops.
He's bad. Well, that's what you get when you don't do the job right. Then this virus does not deserve to infect monocytes, clearly. Okay, so for my second paper today, I'm staying with T cells, moving away from viruses, and talking about a nice publication in Cell Reports from the lab of Catherine Wu at Harvard. First authors, Pavan Bakiredi and Elham Azizi. The paper is called Mapping the Evolution of T cell states during Response and Resistance to Adoptive Cellular Therapy. And it's a nice paper that takes advantage of a study from the 90s. Apparently, clearly they had a lot of samples left from the study, and they decided to use it as a proof, or kind of a model to study, try to identify characteristics of T cells that associate with a kind of immune therapy potential. So let me just explain a little bit. They take on samples from a study of allogeneic stem cell transplantation for patients with chronic myelogenous leukemia. These people get a stem cell treatment, so bone marrow transplant, which is CD8 depleted. So CD8s are depleted from these transplants, usually because they can mediate graft versus host disease, and therefore you take this out to reduce toxicity of the transplantation. And but you still have, for example, CD4 T cells and other immune cells can still be included in this bone marrow. And they basically, patients after this treatment, which was mostly done in the 90s, afterwards, there's other newer treatments, tyrosine inhibitors. They're more popular now. But from these patients, so they have a set of 12 patients, 6 of which were long-term responders and 6 of which were non-responders. So the tumor in their bone marrow relapsed. And they're trying to identify from the T cell response that is kind of pre-existing in these patients and kind of after treatment observed, if they can identify characteristics that differentiate responders from non-responders. So very straightforward. They do single cell transcriptomics, bulk. They also do chromatin accessibility profiles. They look at the T cell clonality of the bone marrow biopsies to look at the different clones of T cells found there. And they find that a couple of things. So again, they look at the single cell RNA-seq, and they generate clusters of different types of cells that it can be found in the bone marrow before and after treatment. And they identify certain clusters that associate with responders, not responders. This paper does a really deep dive into bioinformatic kind of algorithms and bioinformatic models to identify this cluster. It's not very straightforward. So the authors do show that they need to do a couple of normalizations and some take into consideration the variability between the patients. but they find a model which allows them to identify differences between responders and non-responders. So bioinformaticians, if you want to take a look and give me and send me some tips, let me know. And they do find that the clusters that are, and this is not very surprising, right? The clusters that are associated with response are mostly associated with T cell activation and what is traditionally considered T cell exhaustion. And the non-responder clusters are more associated with a variety of dysfunctional states, such as, and have gene clusters associated with hypoxia, with anergy. And so it does seem that they cannot identify a specific dysfunction signature, but more of kind of a diverse reasons that can drive a dysfunction in the T cells of non-responders. What they see is that they identify what are these kind of have previously been described as the T exhausted and the T precursor exhausted cell signatures that are found, were initially described in murine models of exhaustion against viral infection. And again, they find that they can identify this precursor exhausted and exhausted cells in the different clusters, and that responders scored highest for the precursor exhausted that are mostly considered to be able to generate new effector populations upon some kind of immunological stimuli, either antigen exposure or immunotherapy, checkpoint inhibition, or in this case, the introduction of new immune cells that can detect and can respond against the tumor antigens from the leukemia. So they also do analysis from the histone chromatin accessibility, and they generate a model that in which they identify transcription factors and networks of expression. And there's really nice, worth take a look. They can identify specific clusters of expression that are associated with specific transcription factors. And again, they identify transcription factors that are known to correlate with kind of good, productive T cell responses. What they see is that they find that the T cell responses that are characterizing these patients that are responding actually come, most of the CD8 T cells come from the patient that, and not from the graft, from the transfused cells. They find that what they conclude very nicely is that the cells that had been transfused from the donor to the host are not the ones mediating the graft versus leukemia response in this patient, but they're actually reinvigorating the pre-existing response in the patient, mainly by, for example, adding CD4 cells that can co-stimulate CD8 cells, and adding other immune cells that can mediate more immune activation. And that this really, the capacity of the host cells to reactivate upon the addition of the new cells determines the outcome of the patient in this model. Very nice work. A lot of bioinformatics for those looking into that. And yeah, it was very, very interesting to read.
The fact that it's the host T cells or the CD8 cells is really interesting. That the transplant's really just in reinvigorating them is fascinating. I mean, that's what immune therapy is, right? In a way, without the stem cell transplant.
Was really, I also really like that conclusion. I hadn't thought about it, but I think that makes a lot of sense.
No, that's a big one. That's a big conclusion there. All right. Well, we'd love to keep talking papers, and we're going to be speaking to Dr. Phan at the Garvan Institute of Medical Research in the University of New South Wales in just a moment. But before we get to that,
are you interested in differentiating human pluripotent stem cells into monocytes? The STEMDiff monocyte kit from STEMCELL Technologies generates millions of monocytes, ready for downstream assays or further development into macrophages or dendritic cells. Learn more at stemcell.com/monocyte kit. Speaking to us today from several time zones away is Dr. Phan, who is a Senior Principal Research Fellow at the Garvan Institute of Medical Research at the University of New South Wales in Australia, where he heads the Intravital Microscopy and Gene Expression Lab. Dr. Phan, it's great to have you here with us today. Greetings to the future, to tomorrow over there.
Hi, how you going?
Yeah, this is like we had one other guest from Australia, but this is the first time we've done it where we've had Brenda in the evening, myself in the afternoon, and our guest in the morning. I don't think we've done this before. So you're making podcast history here for pan worldwide time zone spanning days and times everywhere.
Well, it's a very exciting times.
It is. Brought to you by the power of everyone being virtual. So thank you for being here today. I was super excited to get to talk to you because I'm also a glorified microscope nerd, but I come from the land of single molecule biophysics in my past life, 2 past lives ago. And so I was very excited to see, your innovative work and approaches using two-photon microscopy. So I was hoping we could start there. But since when I, start counting large numbers, like 2, no, sorry, just kidding. When I start saying photon microscopy and intravital microscopy, I think some people know how it works, but not others. And particularly I was hoping you could explain to, the scientific crowd here. We have a pretty scientifically literate audience since it's usually mostly, scientists listening. But explain how it works and why, in particular, it's two-photon, because I think that's part of the key to the technology that gets lost. Like, oh yeah, two-photon is kind of a fancy microscope, but why you need two-photon for intravital and how it works and what you're able to do with it because it works.
Yeah, okay, cool. I mean, I guess from your work with single molecule, you would appreciate that one of the goals is to overcome this diffraction limit, which is a physical law essentially that limits how well resolved you can image an object. So intravital microscopy, what we're really trying to achieve is minimally invasive imaging in a live animal. So we want to see the physiology in its true biological context. So that means that we want to overcome a different physical law, which is the depth limit, how deep you can penetrate into the tissue without destroying it. And two-photon allows us to do this essentially because it allows us to image in the near infrared range. And in that range of light, there's a natural optical window so that light can penetrate much deeper. That's essentially the reason why two-photon is so good for deep tissue intravital imaging. The other advantage, I guess, is that essentially what we're doing is fluorescence microscopy, but instead of using one photon to excite the fluorescent molecule, we're using 2 photons. And because we're using 2 photons, then each photon is half the amount of energy. And that means that there's less tissue damage. And that allows us to perform volumetric imaging over time. So we can achieve time-lapse imaging. And so what we get is very complex 4-dimensional image of the biology in question.
Can you maybe introduce also our listeners to the type of research you can only do with this kind of techniques, and that you have some recent publication and your lab has been working on? Maybe introduce us to the research that you do with these techniques.
So for us, intravital microscopy is a very powerful tool for biological discovery because it's allowing us to see processes in live animals that we wouldn't normally necessarily be able to see. A really good example of that is some recent work that was done in which we looked inside the bone in a live animal to look at the behavior of this really interesting cell called the osteoclast. Now, this is a cell that's formed by the fusion of monocyte macrophage precursors, and it's the cell that resorbs bone. So a bone is constantly being remodeled in real time. So this is a very dynamic, ongoing process in which the body responds to the environment, for example, weight bearing, and so constantly new bone is being made, and then old bone is being resorbed. And that's the function of these osteoclasts. And previously, because bone is such an opaque, thick, mineralized tissue, the only imaging that we could do in the bone was destructive imaging, in which the bone is harvested from an animal or post-mortem, and then imaged under a light microscope. So using intravital microscopy, we saw that these osteoclasts, when they resorb bone, they weren't just formed from the fusion of these precursors, but the osteoclast itself can also break up, fission into daughter cells. And then these daughter cells, which we called osteomorphs, could also migrate towards each other and fuse to reform osteoclasts. So that's an incredibly dynamic process that we never would have been able to encounter from the two-dimensional static imaging of the past.
So I want to jump on that osteomorph. I know I'm going to go back to two-photon in a second because I think there's a technical question to explore with people. But that's my microscope nerd coming. But before I get there, with this osteomorph, I was reading that paper and really trying to, I'm not a bone guy, but I like osteoclasts because they're macrophages. But going from there, how would you describe this osteomorph? Because you basically found a new cell type that was not really appreciated before through this really cool technology where you could actually just see it happening, which lets you really question what else is going on in our bodies that we're not looking at yet. But what would you describe its function? And then furthermore, do you think there's then parallels based on what you saw, given that it is a macrophage-esque lineage? Do you think there's any other cells that have morphs that we don't know about that you're hunting for next? Maybe if you can give us a preview.
Yeah, no, sure. I mean, look, I think that's a really interesting question. For us, something that we're trying to grapple with at the moment is the question, why? Why would an osteoclast essentially fission and then recycle? I guess our first stepping point was the fact that conventionally osteoclasts have been thought to have a very short half-life, and that when they finish resorbing bone, they undergo apoptosis and die. Of course, that was recently shown in very elegant parabiosis experiments by Frederick Geisman to be not the case, that they are in fact more longer lived than we considered. So one of the consequences of being able to recycle is that you could potentially be able to generate cells that would live for much longer, and whether they retain some sort of functional specialization that allows them to more efficiently form osteoclasts again is one possibility. I guess the other possibility we're grappling with is the idea that actually osteoclasts are incredibly metabolically active. They have to form this very tight sealing zone on the bone where they pump huge amounts of acid and degradative enzymes into that resorption pit to degrade all the collagen and mineralized matrix within the bone. And that probably creates a lot of cellular stress. And we're sort of wondering now with the idea that maybe the cell fission is a response to cellular stress, and it is a part of a mechanism for the osteoclast to resolve the accumulating oxidative damage and so on. So these are sort of areas of very active research in the lab, and we're hoping that we might get some answers soon.
On that note, maybe a more philosophical question, but how do you see the kind of the original knowledge that is derived from in vitro experiments, for example, especially in the case of these cells that are so dependent on the environment in which they find themselves naturally? How much do you think having the advantage of using this intravital microscopy, of intraviolet imaging, will change what we know about certain cell types? And maybe you want to tell us, where did the idea come from that the osteoclasts were short-lived? Why did we think that before, and now you show that that's probably not the case?
Yeah, look, I think in the case of the osteoclast, the bulk of our knowledge has come from in vitro cultures. One of the biggest breakthroughs in osteoclasts and bone biology was when investigators learnt how to culture macrophages and get them to fuse to form osteoclasts in vitro. And of course, under in vitro conditions, we could see that osteoclasts were short-lived. And from that was born the idea that they are short-lived cells. One of the problems, of course, with in vitro cultures is, one, you're not completely capturing the entire in vivo environment. There are lots of very, very subtle interactions, a lot of interactions at a distance that we're only just beginning to unravel, and a lot of unexpected interactions as well. I mean, I was just marveling this morning at a new paper describing, how the brain remembers inflammation that's going on in the gut. So for us, I guess, the value of Intravital is that it makes no assumptions. What you see is exactly what is happening. Of course, one of the other areas that I have a sort of an opinion on, I guess, is that a lot of people are really excited nowadays about 3D cultures and organic typic cultures, and that's great as well. But to our thinking, those are still very much in vitro systems in which they're only as good as the inputs that you put into them. In other words, the rules that govern the behavior of cells within that very limited three-dimensional space that you've created. So going in vivo allows us to see all these things in all its glory, essentially. And I guess the other point I would make, and something that we learned really early on, is that with intravital imaging, particularly two-photon microscopy, which depends on fluorescence, we are trying to make the visible invisible. So we go in there, I guess, with our own biases and assumptions. And so we're trying to see what we want to see. And so we're really careful then to really make sure that, one, what we see is completely reproducible, but also more importantly, that we undertake a rigorous process of cross-validation. So in the case of the osteomorphs, it was very important for us to cross-validate. And that meant being able to show by flow cytometry that we can see these cells, and then by single-cell RNA sequencing to show that the cells were existing in a distinct transcriptional state from the osteoclast and the macrophages from which they came.
Right. So I have a follow-up here before I go back to the two-photon, which I think is an important thing people miss. Do you think there's macromorphs? So you have you have osteomorphs. Do you think there's macromorphs? And I don't know if you can confirm or deny that you're hunting for them, but I would be hunting for them if I found out that osteoclasts had this osteomorph state and they came from macrophages.
Yeah, look, I think that's a really interesting question. Of course, we see this sort of process involving macrophages in granulomas, where particularly in response to a foreign body or to infection with an organism that can't be eradicated, the macrophages fuse to form these multinucleated giant cells. We can also see these multinucleated giant cells in pathological conditions like giant cell tumors in bone, and also some forms of vasculitis called giant cell arteritis. So we definitely know that they exist and they're out there. I think something that you sort of alluded to earlier, which is the possibility that a lot of the rules of engagement, so to speak, that we apply to cells and how they behave might not necessarily be complete, I think is a very good one. We have this idea that cells have this cell wall that separates them from the rest of the outside world, that each cell is unique, kind of like an individual, that we're all unique, and that we retain our self-identity, even though we interact with others. But increasingly, we recognize that we're, in the same way that when we shake hands, we might be transferring information from one individual to the other. Yeah, so it may well be that in vivo, when cells interact with each other, no matter how transiently, it's possible that they are also passing on information in other ways that we're only beginning to guess at. For example, through exosomes and membrane transfer and so forth.
On that, so I just want to ask one very basic question that I probably should have asked earlier in our conversation. But so we have osteoclasts, which are basically macrophages of the bone, to put it in a way. Why do we need macrophages in the bone?
So like Jason, my first love was the B cell. My second love increasingly is the macrophage. And I think they're an incredibly versatile cell. What's really interesting is that it seems that throughout our body, we have dispersed from birth macrophages that reside in the tissues. And depending on the tissues involved, they all have very specialized functions. The one thing that's really interesting about macrophages is that they're very well programmed, I think, under homeostatic conditions to clear away dead and dying tissue in a non-inflammatory way. And that's critical for tissue homeostasis, because if you don't clear away the garbage, so to speak, you get the accumulation of, from an immunologist point of view, you get the accumulation of cytoplasmic and nuclear self-antigens that could trigger an autoimmune response and can lead to disease. And also, I think macrophages are obviously very important in embryogenesis, because as the new limb buds form, etc., the tissue has to be sculpted to form fingers and toes and so forth, and it's the function of the macrophages to clear away this excess tissue. So in the bone, we think the macrophages fuse to form these multinucleated giant cells, because it's clear that the more nuclei an osteoclast has, the more resorptive capacity it has. So one way of thinking about this is that it may well be that it's a mechanism for it to increase its ability to express the proteins that are required for bone resorption. So you can upregulate gene expression, but of course, if you have more copies of the genes, then you can upregulate it by multiples of the number of nuclei that you have. And I think, that's what we think is happening in the fusion process.
Quick follow-up. Are there other types of cells that are composed of fused macrophages? Do you find this multinuclear macrophages in other places besides osteoclasts?
Yeah. So definitely the classic example is when you get infected with tuberculosis, the mycobacteria, mycobacterial infection. The immune system can't eradicate the organism. So what it does is it tries to ward off, and it forms these structures called granulomas. And these granulomas are composed of multinucleated giant cells that have engulfed all these mycobacteria, but haven't been able to degrade them. So we see, again, this concept that the macrophages are fusing because they're unable to degrade something. And so they're trying to increase their degradative capacity. And we also see them in granulomas that form in response to infections that can't be cleared, or in response to inert foreign materials that get trapped in our body. We also see them pathologically in these diseases like giant cell arteritis, which is a vasculitis, inflammation of blood vessels, and in these giant cell tumors that can form in bone. So definitely they're there. And it'll be really interesting if similar rules of engagement apply to them as apply to the osteoclasts and osteomorphs.
Isn't there a similar process in atherosclerosis? So in the foamy macrophages and multinucleated centers occasionally, and large nasty plaques?
Yeah, so you're absolutely right, Jason. And I think what's really interesting, and a lot of people have sort of caught on to where we might be wanting to go, is that when the osteoclasts fission into the osteomorphs, we're able to find cells in the circulation that resemble the osteomorphs. And one idea is that the osteomorphs are able to enter the circulation and recirculate. And so the idea is that they may be able to go to other bones and form osteoclasts there. And of course, one of the questions is, are they involved in other diseases? For example, in rheumatoid arthritis, where osteoclasts are part of this destructive tissue called the pannus, which resorbs and erodes the bone in rheumatoid arthritis. But also there are some interesting similarities between these cells and, as you alluded to, the foamy macrophages in atherosclerosis. And so we're sort of very curious about this.
Yeah, no, I think this is super fascinating. And I think, we're going to see your next paper on what we're calling it, macromorphs, which is an awesome name, soon enough. But to go back before we hop into the other side of the cells here, to your first love, the next question I just wanted to really go back in time a little bit was, what is it? And I know the answer. I'm fishing for it because I think it's really important other people understand when we talk about two-photon. Oh, it's two-photon. That's like two photons. What does that mean? But what is it about two-photon that lets you thusly do it on on living tissue and bypass the diffraction limit?
Yeah, so sorry, two-photon doesn't allow you to overcome the diffraction limit. It allows you to overcome the depth limit. Yeah. And so there's so many advantages of two-photon. We can spend an hour talking about this because I'm just in awe of the technology. But essentially, because you're exciting with two photons of half the energy, that means that the photons are twice the wavelength. So normally for fluorescence microscopy, we're using visible light to excite fluorescence. Because now we're doubling the wavelength, we're moving the excitation wavelength to the near infrared. So because it's a longer wavelength, right, two things happens. One, this moves us into this optical window, this natural optical window in which light can penetrate further. And the reason for that is the major cause of light absorption in tissues is due to things like melanin, lipids, hemoglobin, and those molecules absorb light in the visible spectrum much more than they do in the near infrared wavelength range. So that means that the near infrared laser can penetrate deeper into the tissues. And also because it's a longer wavelength, it can also penetrate deeper. The second thing is that because it's half the energy, then we don't have any problems with phototoxicity. That is where the light hitting the cell damages the cell because it's transferring heat into the cell. And because it's half the energy, there's much less phototoxicity. And that means that we can perform time-lapse imaging. So image the same tissue over and over again over very long periods of time. In our lab, we've imaged as long as 24 hours. Of course, other problems get in the way, but essentially the point here is that we don't have problems much in the way of phototoxicity, but also photobleaching. I think the other thing that is really cool about two-photon microscopy is that you get something for free. So there is this non-linear optical process called second harmonic generation. Now, this is where a molecule that has a certain physical property is able to absorb light and then emit it at half the wavelength. So it's also called frequency doubling. Now, the molecule in biological tissue that does this the most is collagen, and that's because it has this repetitive structural units. So we see it for collagen. We can also see it for other biological molecules like microtubules. But the point is that a lot of the capsule is made up of collagen. The bone is made up of collagen. And so you get this label-free imaging that provides us structural information. And so you get this incredibly rich three-dimensional volumetric image, and then you can image it over time. So essentially you're doing four-dimensional imaging.
So thank you for indulging my nerdiness to help explain to others. This one, my last real follow-up before I hand it back to Brenda is, how the heck do you do imaging for 24 hours on a living creature? That is a lot of anesthesia. Do you guys have shifts with like people coming in and monitoring the mice under isoflurane, which I know is a pain because of the temperature monitoring you have to do? Like, how do you do this for 24 hours straight?
Yeah, so it really is a big challenge. Our biggest concern is actually that the physiological insult that how that impacts on the biology that we're imaging. So we're always super cautious when we do some of those long experiments. But you're absolutely right. The key is the anesthesia. We need really, really stable anesthesia, and we really need to have very careful monitoring. So temperature monitoring in particular is critical because small animals can't maintain their body temperature when they're anesthetized. And you're absolutely right. For some of these experiments, we do have shifts. Monitoring the sugar levels on the scientists is just as important.
And so Coming back to the kind of research that you can do with this technology, you also looked into other types of structures. So besides bones, you also have some really nice research on lymphoid structure, on B cells, and particularly niches within the lymph nodes. Would you like to share with us a little bit about that?
Yeah, no, thank you. So one of the things that we're really keen on as a concept in our lab is the idea that cells are incredibly dynamic, and their behaviors is very much context dependent. And that context is provided by the microenvironment in which they find themselves. And so for us, one of the things that was really interesting was some recent work, and people have seen this in a non-dynamic way before, of course, which is that after vaccination, you do accumulate memory B cells and also memory T cells in the draining lymph node. And there are more memory B cells in the draining lymph node than there are in the non-draining lymph node, suggesting that there's a resident population of memory B cells that stay in the lymph node. And so the niches in which they reside, the cells that they interact with that maintain them in that niche, is something that's really interesting to us. And we see them localizing in this space in the vicinity of the subcapsular sinus macrophages. So there are also lymphatic vascular endothelial cells, marginal reticular cells. So we think those cells together make up a supportive niche that attracts and keeps the resident memory B cells in that location. And what's really interesting is that, of course, when you encounter antigen, it drains the lymph node via the afferent lymphatics. And then the afferent lymphatic drains into the subcapsular sinus. So that is going to be the lymph tissue interface. That is where you're going to first encounter a pathogen when it comes into the lymph node. And so it makes a lot of sense then in the draining lymph node, where you've encountered antigen before, to keep the memory B cells in that location. So for us, that was really interesting. And we showed that also when you reactivate the memory B cells in the draining lymph node, they form this very specific structure in that space, in that subcapsular niche, which we call the subcapsular prolific focus. And that makes a lot of sense also because the macrophages there are specialized to activate B cells because they capture and surface retain their antigen intact. And also we've seen memory T follicular helper cells reside in that niche as well. So this subcapsular niche then can be really important because it provides all the ingredients that you need to generate an antibody response very rapidly on recall of antigen. So one of the other things that we're really curious about, if I can use your question as a segue to introduce something else that we're becoming very excited about, is how do we interpret the behaviors of the cells that we're seeing in vivo? How do we develop a language or a way to capture their behaviors and understand it? And here the problem is one that we always encounter, which is that we look at it through the prism of our lived experiences. So that means that when we analyze the migration of cells within the lymph node, we think of it in terms of the cell, for example, following a chemokine gradient. So that pulls it in one direction, pushes away from another direction. And that's pretty good. And that can tell us a lot of things. But more recently, I guess, we just started wondering whether that's a very limited worldview and whether we can take a more agnostic approach in terms of analyzing cell behaviors. And from that, develop a better understanding of what's guiding them. One example might be to think about each cell as being an agent. And of course, there are lots of agent-based models. And one of the interesting things is that out of the behavior of some very simple rules of migration, you could generate complex, seemingly intelligent emergent behaviors that might then explain how those cells do what they do. And that might then give you a hint to what the underlying biology might be. So for us, looking at how those cells migrate within the follicle and then within the subcapsular niche can also be very informative.
All right. Well, I know Brenda would keep you here talking about B cells well until the night when she passes out, which will be like, 10 a.m. for you. But that being said, we have to wrap it up. And so we always like to end with a couple of fun questions. I'm going to go a little off script since you said we could ask you anything. And I'm going to ask you two very unique ones. The first question is, if you could be any type of immune cell, what would it be and why? And the follow-up is, if you could be any type of microscope, what would you be and why?
Oh gosh. Okay. Well, I love that. I love those questions. Well, look, if I could be any immune cell, I would want to be a B cell. I would especially want to be a B cell right now because everybody in the world knows what a B cell is and what it does. I think we're living in a really, obviously, it's very scary. It's very challenging. But I guess one of the things that's very exciting for me is having everybody know what a B cell is, what a plasma cell is, and why antibodies are important. I think there's an increasing literacy about science and what scientists do, which I think can only be good. So I would want to be a B cell. If I could be any microscope I could be, then I would want to be a microscope that hasn't been invented yet. And for me, I think one of the real exciting possibilities is that with particularly a lot of these so-called nonlinear optical processes, you can achieve label-free contrast imaging without the need, obviously, to have, genetically encoded fluorescent reporters and so forth. And what that opens up is the possibility of doing intravital imaging in live human subjects. And for us, ultimately, I mean, what we're seeing in the mouse is, we're hoping is a mirror of what happens in people, but ultimately what we want to do is understand human biology and human disease. So if you can come up with a microscope that allows me to do that, that's the microscope I would want to be.
So you want to supersize a microscope and make you want to be the supersized human version of your two-photon system to see that collagen light up in a person.
Yeah, it may not be two-photon, it may be a three-photon microscope, for example, which can image label-free contrast between refractive index mismatches and provide you with cellular resolution. It might be a next iteration of, an optical coherent tomography, which we're already using in the clinic. For example, ophthalmologists are using OCT to look in the back of your eye and seeing, cellular resolution inflammation going on the eye. I mean, those are really exciting things. And that's where, getting down to human biology and human disease will be really cool.
That's an
great
awesome
answers. Yeah. Great answers. Thank you. Well, it was such a great conversation. Thank you so much for joining us. For listeners interested in your publication describing osteomorphs, we'll put the link in the show notes. And published in Cell. Congratulations. Very nice. And well, it's been really, really, really great to talk to you.
Thank you so much.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. Suggestions always welcome. See you next time.